what species are in the products and how well do they match the label. And the setup that we are following is that here you can see, for instance, it's a saffron. So it's a product that it's very often, it's an expensive product, so it's quite often forged. So I think it's a good example because people replace saffron with other species quite often. And then what we do is that we do DNA extraction, and the setup is that we generate a number of technical replicates. And we, in our current setup, it's 3 different replicates. We prepare libraries, then we send them for sequencing. I will stick to ion torrent sequencing here for an example, but the whole pipeline is modular, and the modules are interchangeable, so you can, if you change your sequencing platform, you can easily change the module in the pipeline, and I know from ion torrent you can go to analyzing Illumina. And then I will focus on this small server, what is happening inside and how the pipeline has been implemented. So from the bioinformatics point of view, not all the steps are super important. We actually start being concerned about what is necessary for efficiently processing and analyzing the data. At the moment where we prepare libraries, because we want to know what barcodes we are using, because we need to, we analyze samples in batches. So it's quite often that we receive 100 samples and we have to multiplex them in order to sequence them efficiently. And we are also using a number of cell flows sometimes because it's not possible to fit all the replicates and all the samples on one flow cell. And then we are using a very specific sequencing platform, and then we need to know some things, some features about this. So we are concerned about how to trim our reads, what qualities can we expect, what are the specific biases coming from a particular sequencing platform, what yield we can expect, and as I said, like, we are quite often using a number of flow cells, so we need to come up with a really nice scheme for demultiplexing in a reasonable time without avoiding all possible mix-ups and errors. And then we need a pipeline that is robust, reproducible, and that we can easily control and interchange different modules within the pipeline. So there is a number of tools available for designing pipelines. One of them is SnakeMake. SnakeMake is a very popular workflow manager, so it enables you to modularize your pipeline and if you want to rerun only certain steps in the pipeline, it's very easy achievable with SnakeMake. The problem with SnakeMake is that it really fails with demultiplexing because we do 2-stage demultiplexing. First we demultiplex by barcodes, and then we demultiplex by primers because we are actually looking at both ITS1 and ITS2. And SnakeMake cannot handle this 2-step demultiplexing for technical reasons. So we were looking at another workflow manager, Nextflow, so we implemented part of the pipeline there, but we thought it's a bit of an overkill. So what we are currently doing is actually that the part of the pipeline is just a standard bash scripting, and then we outsourced most of the analysis into R, which is my favorite programming language, and that's why partially I decided to go for it. And in R you also have some workflow managers like FlowR, for instance, that you can use. And then we have what is important for us as well. It's a bit slightly different from a standard research project because this food safety authority, it's a different type of user. I mean, they want their data to be presented in a nice way and in a bit less complex way than we usually present the data in articles. So we need some kind of a nice presentation layer, and we are, to this end, we are using Neater package and Shiny application framework to actually create reproducible reports that are automatically generated along with the analysis. And then we also use this Shiny app so that we can, in a nice way, we can have an interface that we and our users can actually move some sliders and see how the result changes and so on. And finally, currently we are running, we have a small server, very simple setup, but if we want to scale up in future, maybe if we want to go for cloud solutions or so, then we need to package everything in a container, for instance, Docker container, so that the whole workflow is portable between different systems, being it your PC or Mac, Amazon Cloud or HPC solution. And then when I think of the general overview of the pipeline or the workflow, then I can identify a number of layers that each of them consists of a number of steps. So first, it's the layer 0. So we obviously need a reference database because we need to identify our species based on something, right? So we need to compile a reference database, and we also need to do some cleaning, and I will elaborate on this a bit later. And then there is a second part of the second layer, which is processing of raw reads. So it's nothing fancy here about it. It's standard. So in this case, ion torrent reads. We are demultiplexing by barcode, then by primers, then doing some preliminary trimming and very preliminary quality control. And these two layers are realized in Bash mainly. And then I'm migrating, the whole workflow starts using R, entirely R, and I'm doing quality control and trimming. And then we are currently in this version of the pipeline that I'm presenting now, which we are using actually to deliver the results. We are using amplicon sequence variants instead of OTUs, so we are using this approach. I don't know how many of you are familiar with this approach of data to how do you... Okay, well, I will try to briefly describe how it works. So basically, you have a number of reads, and from this large number of reads, you can actually infer some probabilities of certain types of errors, different transitions and transversions. And in a way, it's a similar approach to when you are doing whole genome sequencing and you are using base recalibration, for instance. So there is a machine learning algorithm that learns error patterns in your particular run, and then tries to correct those or take into account these probabilities of errors when actually doing clustering. So it's not so super different from the OTU approach. It is just the error correction step that is done prior to clustering. But it allows us to achieve much higher resolution, at least in theory. Then we are doing deduplication and chimera removal using de novo chimera detection, and then we are inferring amplicon sequence variants. And then we are going to the last layer, which is consisting of species identification, and we have this interactive tool that enables us to actually play a little bit with parameters and see how the results are affected. And when we look at the particular layers, so the first layer, which is demultiplexing, so throughout the whole process, we are using this multi-QC tool for actually follow the qualities of the reads and keep track on what's happening. And then for demultiplexing, we have found out that of many available tools for us, FlexBar is the most flexible approach because it allows us to actually do demultiplexing and trimming for not only, it is not fixed for one sequencing technology. And then we need to do this step, which is in red, which is surprisingly very critical, and it's always some problems with this. Because as I said, we quite often are getting data from different flow cells. And then it's like, we see that this is the needs a manual intervention. You always have to manually check that everything went fine. Pretty surprising. I mean, theoretically it's very simple, but in practice it's the step that requires quite a bit of attention and manual curation always. but we're trying to optimize it now to automatize it completely. And then we are doing some trimming using cut-adapt, and we are ending up with demultiplexed FASTA files. So we have a product, we know from which ITS was amplified and which replicate the reads are coming from. And then we are looking at the reads quality. So we randomly select a number of reads completely randomly from the files, just for the sake of computational efficiency. And then we are aggregating the qualities, read qualities, and we can see that these are the plots generated by this data2 or data squared, however you should pronounce it really. So you can see that along the read, you see the green line is the median quality. Then you have some quantiles of the qualities as these dashed lines, orange lines. Then the shade of gray in each of those squares or rectangles corresponds to the actual number of reads that had this particular quality at this particular position. And from these we can just see how the qualities of our reads are in this particular run, and we can adjust trimming if necessary. So, as I said, it was random subset of reads for quality control plots. When we use this information from this step, we are doing trimming on all the reads based on the qualities. And then we proceed to the error learning step. And currently, for example, for Aryan torrent, we found out that we are trimming all the reads that are longer than 360 base pairs because they usually drop. The quality drop is pretty significant after 360-ish. Then we need to also, it's a very Ion torrent specific, but we need to trim from the left 15 base pairs because it's also a lot of contamination there. And also if quality drops at any point in the read drops below 2, then we discard the whole, the rest of the read. And then we are learning errors using, well, in our case, we are using 1 million randomly selected reads. And you can see that without going in very much into details, but here what is shown is that you have consensus quality score on the x-axis. And then you have error frequency. So you can see that, and here each of those plots is particular type of error, so either transition or transversion. So you can see that, for instance, probability of transversion A to C for particular quality class is at this frequency, right? So for your particular run, you actually know what are the probabilities of different types of errors, and you can use it to correct these possible errors to actually reach higher resolution. Once this is done, we do dereplicate our reads, and then we do sample inference and de novo chimera detection, and we proceed to the next step. And this is the species identification and presentation. So we have this ASV table similar to OTU table. So in the rows you have identified amplicon sequence variants, and then you have products as your columns, and then you have a number of reads supporting every ASV per product. And then we are just using a standard BLAST approach, but we are trying to use custom databases. Currently, we are using this ITS2 database that is available from the University of Würzburg. It is a manually curated database that is compiled from different sources. But we have found some issues and challenges there, so we are actually trying to improve this. And then for each and every amplicon sequence variant, we are returning 4 top hits, BLAST hits above 97% identity. Then we we get some identifications, and we spit out a static report from the whole analysis. But also we have this user Shiny dashboard, which I will show you. So we can play a little bit with different parameters and see how they affect the species identifications. And we also try to keep our ASV table and all the downstream analysis. We are following this new paradigm of tidy data and tidyverse, which is like for our people, it's a big thing now, but it's also a very convenient thing to work with. And then we are presenting our results in a few different ways. We began by, so here you see an example for one of the spices, one of the samples. You can see that we are looking at ITS2, 3 different replicates, and here you have different species here and different ASVs on the x-axis. And then the size of the bubble is corresponding to the log number of reads supporting this particular identification. And also if there was only one species identified per particular ASV, it's presented in red. But if it was multiple identifications, then it's in blue. We can also visualize the same as word cloud, because the people from Food Safety Authority like it, because it's like comparing word cloud to the label on the product, you know. It's really cool for them. They don't care much about other parameters because it's a very new thing to them, very new approach. But now we also even simplified our report, so we collapse data a little bit. We don't show particular ASVs, but we actually show just the species and how many replicates support an identification. And we have currently, we say that an identification is true positive if there is at least 2 replicates are supporting this particular identification. And here we have this tool that we can play a little bit with. So let me try to run it. If I'm lucky, then it will run. Okay, so I can open it in my browser. I can load data. So it's a result of blast identifications. And then I can select the product of interest. So say that I will see product 37. It's just a nettle that shows up, but I can also say that I lower the threshold, so the minimum number of replicates is just 1. So you can see now the plot changes, and here you have a tabularized version. And you can play with different parameters here. You can limit the number of top blast hits and so on and so forth. So I think it's a pretty convenient way of actually playing a little bit with the whole workflow and data presentation, and you can also leave it to the user sometimes to change those parameters and see what's happening. You can also compare easily. You can change to ITS1, for instance, and see what ITS1 is saying. And currently, so I'm working on like much more complex interface that will allow you to actually manipulate all the parameters from the very beginning of the workflow. Yes, and now to some real challenges. So we found out that probably the most challenging part is that the databases, reference databases, are poor. We were first using GenBank sequences, and what we actually do is that we have a number of FASTA sequences which we annotate with NCBI taxonomy, and then we do in silico PCR using our primers to see what amplifies from the database. We trim out primers, we collapse duplicates, and then we construct so-called equivalence classes. I will show in 2 slides what I mean by equivalence classes. So we do then also identify challenging sequences, and then we need to do some manual curation, and then we construct a BLAS database from this. But it's pretty challenging actually, because in this manually curated ITS2 database, we found 5,500 problematic, challenging sequences, which just by looking at species label and sequence, so we are comparing, everything is fine if you have different sequences and different identifications for them, but there is also some problematic cases where we have the same sequence with different identifications. Sometimes you have situations that here you have a lot of species from the Draba family, but then you also see some typos. So Draba became Abdra, somebody misspelled, and there's quite often the source of problems with those reference databases. And there is surprisingly many sequences that have same ID, sequence IDs, and so on and so forth. So you can even go farther, and you can see that you can have a whole bunch of 8 different classes of problems based on whether the species have same annotations or different annotations, and then comparing sequences and accession numbers. We can address most of those problems, but not all of them and not always. But what we are currently doing is that, as I said, we are constructing those equivalence classes. So I can see if there is identical or super similar sequences in the database, then I'm basically merging them into one equivalence class. So then I'm getting my identification, and I can say that it's either of those 2 species. So it's still higher resolution than the family level, but at least I'm honest, and I see what are that there is some problem with identification. I cannot do better than this. And few words about how does it scale, like how much time does it take? Because as I said, we need to deliver really quickly and a reproducible and reliable result to this food safety authority. So it's pretty stressful. We are receiving a batch, and then we have few days to actually give them the final report. And we want to avoid sleepless nights and so on. So we want everything to be running reasonably quickly and to be streamlined. But fortunately for us, it's no big computational bottlenecks here. So we are using this very simple machine, 8 cores, 2 gigabytes of RAM per core. And we found out that demultiplexing is the most time-consuming step. It's about 3 hours for 300 or 100 samples, but 3 replicates, so 300 samples. Then ASV inference, I can do it on my computer. It scales linearly, or in the worst case, it's some logarithmic complexity. Database curation is a bit more time-consuming, but still not bad. And then what is the most time-consuming step is that when we are preparing these custom databases, it's the in silico. In silico, it's my spell checker. So again, but it should be in silico. In silico PCR is pretty time consuming, but fortunately we don't do it for every run of the workflow, but we do it like every few months, right? So to update the references and finally, like what are our plans for the nearest future and what we are trying to do to make it working better? So obviously better custom databases. I think it's the biggest bottleneck for us, at least in the plants world, because it's really a lot of species with wrong identifications because the taxonomist could not identify correctly voucher from herbarium or so, many reasons. We are building our own databases because there is a lot of local variation as well. There are cultivars, there are local varieties that are different between like even different parts of the country sometimes. We would like to go for the machine learning and both for species identification, and I'm planning to use some technique that is called rough sets. In short, the strength of the method is that it is very honest about how ambiguous an identification is, so it can actually give you some either probabilities, or it can say like, based on the data, I cannot really identify this sample. Then it's an interesting approach that I have started working with on the plane here actually, and it's to skip the whole alignment and blasting and just use your reference database and use a deep learning algorithm, most likely convolutional neural network. So basically, you teach the neural network on your reference database, and then you feed it with your reads, and then it identifies everything based on features that we have no idea about because it works like a black box. But given enough examples, people claim that it works really well. It works really well for image recognition. I'm not that sure about sequences, but so far it seems to be promising. So maybe a way to go and test this. We want to have modules for all, or at least the most common sequencing platforms. We want to improve the parameter optimization and automatize it a little bit so that if runs differ between each other, you can tweak the pipeline a little bit. Yeah, and working on this Docker container for portability as well. So that's the things that we are currently working with, and yeah, we are using this TensorFlow, Google's algorithm for this step actually. So that's why the picture is here. And I would like to finally mention that it's collaboration with Hugo, but also it's my colleague Nima Rafati who is involved quite a lot in the bioinformatics part. And I would like to thank you for your attention. And
session is open for discussion.
Yes, so I think that the idea to use machine learning algorithms to identify species is very interesting. Do you know if it has been done before? Not for plants, but for other kinds of species?
So I have seen on bioRxiv, I have seen a paper actually discussing this. So they are using something that is called database neural networks, and they claim that it works really well. It is an interesting point because the whole philosophy of using machine learning in this case would be that there are some properties of sequences that are hidden to us, that they will be artificially derived from sequences, and they will be unique to different species, which is tempting from one point of view, but I think that we need sufficiently long stretches of sequences to actually make it work.
And haven't you tried using gamer profiles? Because they are also alignment free. Okay, you have to build first the reference. Mm-hmm. That's the same as building the neural network. And do you know if they work well in your data? Because
no, this I haven't tried. What I did, I was trying with these filters similar to convolutional neural network. A pretty naive approach, basically following the documentation, but it seemed to. I was not really trying it on the biological sequences. I was playing a little bit with some strings that I created, and it seems to work.
For bacteria, for example, like they would divide the sequence into, I don't know, eight mers or something like that, that it would work fairly well for taxonomic assignment, but I don't know if that's true for plants too or not.
Definitely worth testing, so thanks for the idea. Right. If there is no more questions, I just still a moment. I would like to welcome you. I'm organizing summer school in advanced bioinformatics using R. So this year it was the first time. It was on beautiful island of Gotland in Sweden. And next year it will be happening. So follow the scilife.se, scilifelab.se education here. And then on courses and training, you can access actually information. So everyone is more than welcome. It's a lot of fun. So thank you.